In this video clip, I will show you two great examples of using fluorescent molecules for life science. Before I do that, I would like to show you the molecules generally called dark quenchers. A dark quencher is a molecule that sucks energy from a fluorescent molecule and releases the energy as heat. With micromolar or less concentration of the dark quencher, the heat from the dark quencher is too small to affect the temperature of the solution. When the two molecules are close together, fluorescent molecules and dark quencher, emission from fluorescent molecule is almost so completely quenched. In other words, the quenching effect is distance dependent. Then what you can see here is the chemical structure of dark quenchers. BHQ stands for black hole quencher. The black hole quenchers are capable of quenching across the entire visible spectrum. On the other hand, Dove cell, right here, absorbs the green light only. How can you use dark quenchers? I'll show you an example. One great example would be the technology called molecular beacons. Molecular beacons are hairpin-shaped oligonucleotide hybridization probes that can report the presence of specific DNA sequence. A typical molecular beacon is 25 nucleotide long, and the middle 15 nucleotides do not base pair with one another. At the 5' end of the molecular beacon, a fluorescent dye is covalently attached. For example, Cy3 in this particular design. To the other end is covalently coupled with dark quencher. For example, BHQ2 in this particular case. The beacon itself is not fluorescent because Cy3 and BHQ2 are close to each other. In the presence of the DNA complementary to the loop, the hybridization occurs. The resulting duplex is more stable than the hairpin structure because the duplex involves more base pairs. The duplex formation separates Cy3 and the quencher, restoring the fluorescence from Cy3. That's why you can see the fluorescence from the Cy3 here. This technology allows us to detect the target DNA sequence in the sample. Very nice idea. Let me tell you about the other technology, which was developed by Professor Roger Chen and co-workers at University of California, San Diego. Professor Roger Chen is a well-recognized scientist in the field of fluorescent molecular imaging. He was awarded the 2008 Nobel Prize in Chemistry for the discovery and development of the green fluorescent protein, GFP, which I will talk about later. One day, Professor Chen and co-workers came up with a nice idea to track the location of proteins inside cells. The technology is called FLASH. It was known that arsenic compounds bind to pairs of cysteine residues. Any arsenic molecule can bind to the cysteine SH group, like this. Then that this is the reason why many arsenic compounds are toxic in the body. The research group of Professor Chen synthesized an arsenic compound called Fresh EDT2. It binds to the proteins containing tetracysteine motif. Cysteine, cysteine, something else, cysteine, and cysteine. Upon binding, the probe becomes fluorescent. This tetracysteine motif, right here, would align the 4 cysteine side chains on the same side in alpha helix-like conformation, which allows the first reaction with the flash probe, like this. This technology permits the specific labeling of proteins with tetracysteine motif in living cells. When you transfect a gene encoding a tetracysteine containing protein, the flash probe, which is cell permeable, can visualize the protein inside the live cells. Here you can see the fluorescence in the cells, but if you do not transfect such a gene, you don't see any flash labeled protein. The fluorescent molecules are quite useful in monitoring biological processes. Can you come up with more ideas?